Welcome to this brief module on simulating biological systems, where we'll explore how computational models help us understand living processes. Over the next slides, we'll touch on motivations, common model types, practical workflows, case studies, and future directions, all packaged for quick learning and practical insight into how simulations accelerate biological discovery. This deck covers 5 key areas: why we simulate biological systems, the main types of models, essential steps in a simulation workflow, illustrative case studies, and a look ahead to future platforms and ethics. Think of this as a roadmap that links motivation to methods and real-world impact, so you can follow the story from concept to application. Why simulate? At a high level, simulations let us explore hypotheses faster, cheaper, and more ethically than traditional experiments. They create safe, controllable environments to test scenarios, reveal mechanisms, and prioritize experiments, so researchers can focus limited wet lab resources on the most promising leads and speed up translation to treatments. Virtual experiments bring 2 powerful advantages. They reduce cost and ethical concerns by limiting unnecessary animal or human trials, and they accelerate discovery by screening many parameter sets and predicting emergent behaviors. In practice, simulations help narrow down hypotheses and design targeted wet lab tests, making the whole research cycle more efficient and focused. Moving from reductionism to prediction means integrating data across scales, from molecules to tissues, and building predictive frameworks that forecast responses and guide interventions. By combining diverse data types into unified models, simulations shift biology toward quantitative prediction, enabling faster discoveries and more targeted experiments that would be hard to plan by intuition alone. In the next section, we'll look at model types, from mechanistic equations to agent-based and network approaches. Choosing the right model depends on your question, available data, and computational budget, so we'll compare strengths and trade-offs to help you pick a suitable framework for different biological problems. Deterministic models use equations to produce fixed trajectories given initial conditions, while stochastic models capture biological noise and variability, offering a more realistic picture when randomness matters. The choice affects parameter identifiability and computational cost. Deterministic approaches are cheaper and precise, whereas stochastic methods demand more resources but reveal variability and deeper system behavior. Agent-based models build behavior from the bottom up by encoding rules for individual cells or agents, which then interact to produce tissue-level patterns. This flexibility is ideal for studying spatial heterogeneity, tumor infiltration, or immune cell dynamics, and these models can be calibrated with single-cell or imaging data to explore emergent phenomena that aggregate methods might miss. Network dynamics approaches capture interactions without requiring detailed kinetics. Boolean and Bayesian networks are great for rapid screening of gene or protein interactions and drug perturbations when kinetic data are scarce, while constraint-based models focus on metabolic fluxes and can predict pathway usage under different conditions, often integrating experimental data for refinement. Now we turn to workflow essentials, the practical steps you need for robust simulations. A reliable workflow spans data collection and pre-processing, parameter estimation, validation, and iterative refinement, and each step demands attention to standards, reproducibility, and noise control to ensure models are meaningful and useful. Integrating multiomics, imaging, and clinical datasets poses real challenges due to different resolutions, formats, and batch effects. Adopting FAIR standards helps make data findable and reusable, while pre-processing like batch effect correction is essential so models learn biology instead of artifacts. Harmonized ontologies and careful pipelines turn messy data into simulation-ready inputs. Parameter estimation draws on multiple approaches. Frequentist methods like least squares provide point estimates, Bayesian techniques offer probabilistic posteriors and uncertainty quantification, machine learning surrogates speed up expensive models, and identifiability analysis tells you which parameters can be reliably estimated from your data, guiding experimental design and interpretation. Validation workflows should be tiered and rigorous, combining internal consistency checks, cross-dataset predictions, and prospective experimental tests when possible. Metrics like concordance correlation and calibration slopes help quantify performance, ensuring models are reproducible, accurate, and informative enough to guide the next round of experiments or clinical decisions. In the case studies section, we'll see simulations applied to heart rhythms and tumor microenvironments, showing how computational models translate to clinical and experimental impact. These examples highlight personalization, spatial modeling, and the path from simulation predictions to validation and trials. Personalized heart models use ion channel equations informed by MRI and ECG data to predict arrhythmia risk more accurately than population metrics, guiding ablation strategies and reducing invasive procedures. By integrating anatomy and physiology, these models demonstrated clear translational impact and helped clinicians tailor interventions to individual patients. For the tumor microenvironment, we'll examine an agent-based simulation that coupled nutrient diffusion with mutant cell proliferation to forecast immunotherapy response. The model revealed spatial barriers to T-cell infiltration, was validated in mouse experiments, and informed combination therapy protocols that are moving toward clinical testing, illustrating a full translational pipeline. Looking ahead, the field is moving toward hybrid, explainable platforms that combine mechanistic models with machine learning, while emphasizing uncertainty quantification and ethical standards. The goal is reliable, interpretable tools that can serve as patient digital twins and clinical decision aids without sacrificing scientific rigor or safety. Next-generation hybrid platforms will fuse mechanistic equations and deep learning to uncover missing interactions and enable real-time patient modeling, while explainable outputs are essential for clinical adoption. Equally important is quantified uncertainty so providers can trust predictions, and a community-driven balance between innovation and safety as these tools move toward practice. Ethics and reproducibility must underpin simulation work. Open code, data anonymization, and transparent practices protect privacy and enable independent verification, while bias audits help prevent disparities in AI-driven predictions. Together, these measures build public and clinical trust and ensure simulation technologies are both responsible and reliable. Thank you for following this overview of simulating biological systems. We've covered motivations, model choices, workflow essentials, case studies, and future directions, and I hope you're leaving with a clearer sense of how simulations can accelerate research and translate into real-world impact.